Hello, everyone. This is Debbie Spoons, NSS Webinar Chair, and welcome to CaveNet, our NSS webinar series. I want to thank our presenter tonight, Dave Culver, for being willing to share his education, his knowledge, his experience and skills with our members. We want to thank our NSS members for attending and our parent organization, NSS, for sponsoring this. I'm going to read about Dave. He has a very interesting background, and I also will have him, if he doesn't mind, say a few words about himself, because I've read his bio and it's really amazing. So we're going to go ahead and get going right here. I can get my computer working. Okay, Dave began his caving and scientific career studies under Ken Christensen at Grinnell College, leading expert in Columbia, which are tiny wingless insects that are common in many caves. After he finished his PhD at, under Tom Poulsen at Yale University, got some pretty good names here, in the 70s. He's been studying cave-related life ever since. He studied a variety of topics, including ecology, evolution, biogeography, and conservation, initially in West Virginia caves, which are awesome caves back there, and later in caves throughout the U.S. and Europe. He has a special fondness for I had this right, and amphipods, but he grew to appreciate beetles, fish, etc. Now, he's authored over 100 papers and books on caves. The guy is just amazing. He has a lot of great information for us, and I want him to go ahead and take over. Let me make sure he's unmuted. All right, Dave, you're unmuted. Go ahead, and this is Dave Culver. Go ahead and give us this interesting webinar. Thanks, Dave.
Thanks, Debbie. Thanks for inviting me and giving me the chance to do this. Like a lot of biologists that study cave animals, we're fascinated by their morphology. You can see on this slide some of the basic features of a cave animal. This is many of you probably know, this is an amphipod in the genus Ecological, and the late John Holsinger was the expert on this and described over 100 species from caves. They all pretty much look the same, and their characteristics include the lack of eyes and no pigment. In fact, this organism is partly transparent, which is sort of partly why you see sort of a confusing mess in the body of the animal. And you can't really tell this without a comparison, but also the antennae are longer than you would find in surface dwelling species. And this makes sense because they got to find and sense stuff, mates, enemies, food, all that good stuff. Well, I lied to you about where this isn't a cave animal at all. And in fact, it was collected in this habitat here. You can see my longtime colleague Tanya Pepon and I looking through wet leaves, basically, which is where we find them. This is a small, we usually call them seeps or seepage springs, that's basically emerging from very shallow depths. And we find the animals just in and amongst the leaves. This represents the height of the technology we have to find these things. We've never found any other way to do this except to just poke through the leaves. The reason that these habitats exist is because there's a clay layer underneath that prevents the water from sinking down. And typically they dry up in the summer, or at least there's no running water. The organisms, as far as we know, simply burrow into the clay and wait for better times, that is, the fall rains. Well, in case you thought that this was just a fluke. That is, we have this one odd population. Here is a map of the distribution of this species in the mid-Atlantic region. There are a few other records to the north, but this actually encompasses most of its range. So we're basically in the Potomac drainage and around the Chesapeake Bay. Most of you probably know that there really aren't any caves. If I showed you all of the caves on this map, as far as I know, there are one or two in the very far upper left corner of this, and some small limestone bands east of the Blue Ridge Mountains. But basically, this is Piedmont and coastal plain, and we find this species here. These are not caves. Among the strangest habitats that anybody's found it in, it was basically in sandy areas at the tip of the eastern shore of the Chesapeake Bay, and it was collected in a flooded pitfall trap. Pitfall traps being usually used for terrestrial animals. So these organisms are living in the interstices of this habitat. Well, it's not the only species that we find in this kind of habitat, and here's a list of the species in the general Washington, D.C. area that are found in these seeps and have no eyes or pigment. And there are 6 species of amphipods and 1 species of isopod without eyes or pigment. And there's another isopod and a snail with reduced pigment and reduced eyes. So this is not some fluke, some oddball thing, but rather a very common occurrence of species, and it's something that's widespread and needs some sort of explanation. What we see here is a sketch originally done by Mestrow in 1962 of a habitat that he called the Hypotelminorheic. I have to tell you that word is just one of my favorite words of all time, because it takes some practice to even say it, and it seems to make absolutely no sense. He was Croatian. He was writing in French, and the root of the word is in Greek, and it basically means water flowing under rocks and dirt, more or less. So what you see here is a picture of the habitat itself, not the spring, that's where we collect them, but the habitat, which is between this impermeable layer, which you can see on the sketch, and the surface, which is represented by those wonderful trees that he drew. And you can see what he calls the upper spring and the secondary spring. Those are the sites where, excuse me, you can collect these animals. It's perhaps if we think about a Croatian using Greek and writing in French, we can forgive him for this tongue-twisting word, but it makes it sound really scientific, if nothing else. So this Hypotelminorheic, which we see pictured here, was originally described from the Medvednica Mountains in Croatia, and this site is actually very close to a ski area, so there's considerable slope to this, very different than the areas around Washington, D.C. There are no ski areas, believe me, around Washington, D.C., and there are no mountains. But the basic structure is the same. That is, there's an underlying impermeable layer, and then maybe 1 meter or 2 meters of humus and crumbled stone, and this is the primary habitat where these animals occur. So I start with this as the non-cave habitat because in some ways this is the most extreme of the ones that we know. The animals never encounter light, for the most part, unless they're in those seeps that is in the habitat itself. So it's a dark habitat, but they're never deep. They're never more than a meter or two from the surface. Well, I want to talk now for a few minutes about other non-cave subterranean habitats. And here's just a list of the papers that sort of introduce these habitats, and I'll talk about each one of the habitats briefly. You will notice that particularly the early papers are not in English. So and I'll talk about this later, but most of this work has been done by Europeans. Well, we'll start with somebody that is greatly revered in Europe, Emil Rakovica. Rakovica is a famous Romanian biologist. He was on the first expedition to Antarctica that overwintered on the Belgica. So he has a distinguished record as a real explorer. He wrote this paper, an essay on biospeleological problems, when he was quite a young man. This is not the result of years and years of study, but rather a thorough reading of the literature and a lot of field work. And he decided that most people were just wrong about what they were saying about the caves. So in the beginning of this essay, he talks about how the important habitat in caves are not the cave passages themselves, but narrow cracks not accessible to humans. So he sort of sets the stage and then goes on in this rather lengthy essay, over 100 pages, to talk about a variety of other non-cave habitats where you can find organisms that have no eyes and no pigment. Now, I think the Europeans go overboard in their sort of admiration for him, but he certainly was prescient in a lot of the things that he said, particularly in terms of the ecological situation for organisms that live in subterranean habitats. He also, at the time that he was writing, was at least looked at a variety of different reasons why organisms lost their eyes, including natural selection. At a time when even Darwin, well, just before this, Darwin is actually not convinced that natural selection was involved. He has two other really remarkable characteristics. One is an astonishingly good mustache. This picture doesn't do justice to it, but it's sort of a famous mustache. And the other thing is, and now there's an English translation of this essay, which I recommend as reading for you. And he's a master of invective. I mean, without being really nasty, he can be very, very funny, and it makes for good reading. He talks about some authors giving him a headache because they change their mind in the course of a single sentence. So he spares no one in his wit, in his sharp wit. He's also master of other phrases as well. Another famous phrase in cave biology that he coined was that cave animals are predators by predilection, but omnivores by necessity. So you get a sense of that he was a very vivid writer. But one of the things that he did was to say we must focus not just on caves, but on all of these other aphotic habitats, that is, habitats without light. Well, the first of these habitats that I want to talk about is the soil. And this is the most common. I mean, after all, soil is everywhere. And soil is a subterranean habitat in the sense that it has specialized organisms and there's no light. Light disappears in the soil, within an inch or a couple of centimeters in most circumstances. And Culver, who worked in a lab where people studied cave animals, was one of the few people that really looked at the soil not as just a habitat where plants grew, where soil fertility was important, but rather as a habitat that harbored a set of species that were specially adapted to the soil. And in fact, the soil harbors a number of eyeless, de-pigmented species. You can, almost any place in the world, you can find these animals, including collembola and mites. What I've shown here in this picture, this drawing, is it looks kind of like a worm or a nematode, and it's actually a mite. And if you look at it carefully, you can see the little appendages up there at the top, and everything is just elongated, and it's clearly adapted to slithering through a habitat which consists of the spaces between grains of dirt. So this is a mite trying to become a nematode, I guess is the best way to look at that. So most people that study the soil aren't really interested in this connection to the subterranean world, but in fact, it is a aphytic habitat with a large number of eyeless species. Not all of them are so bizarre and extreme as the one I'm shown here, which is perhaps the most bizarre of the ones that we know. Let me just say that this organism is about 2.5 millimeters long, so it's quite small. It's nematode size, maybe a little bigger than a nematode, and about 0.05 millimeters wide. So it really is like a worm. Well, another habitat, which was first named by another Romanian, Orgadon, and this is the Hyporheic habitat, or he calls it the Hyporheic biotope. And it is simply the underflow of streams and rivers. So if you think about any sort of gravel bottom stream in particular, and if you dig down a few centimeters, a few inches, then you come to a habitat that is also without light and also often harbors animals without eyes or pigment. And it's one thing to say that habitat is there. It was quite another thing to actually sample it. For a long time, people did what they called dig chapuis holes. Well, a chapuis hole is just a hole you dig on the bank of the stream until it starts to fill with water. And then as it fills with water, you extract the water and filter it to find the organism. It works, but it's not a very good device. What you can see here is actually the plans and I'm standing there using one, what's called a bout rouge pump, named for two French biologists. And basically what it is an iron pipe with a pointed end, and there are holes in the end, and you smash that down using a sledgehammer into the bed of the stream. Usually, well, it depends, but usually as deep as you can manage to get it. And then you prime the pump. It's just a hand pump on top, and you prime that pump. And then after you do some vigorous pumping, a slurry of mud and water comes out. And this is the water from the Hyporheic. If that's then filtered through a plankton net, then you can find a number of organisms. They tend to be very small. Copepods dominate this fauna, but you also find isopods and amphipods, typically less than, say, 3 millimeters long, but they can be incredibly abundant. The standard measure to use is 10 liters of water to pump, and then that is sorted through to look for animals. This is an example of a very simple technology that really revolutionized the study of this habitat. Now, it's important not only because, from my perspective, because it has animals without eyes or pigment. It's also important because it has a lot to do with the health of streams and rivers, because there's a constant exchange of water and nutrients and minerals between the riverbed and the river. So if you want to look at pollution and want to look at river health, you have to look also at the Hyporheic of rivers. And in fact, my late colleague Janine Joubert at University of Lyon did this extensively in the French rivers. And there was enough economic interest that she could also support work on caves, which is rarely of any economic concern to anyone. Well, the next habitat I want to show you is one that's called the milieu souterrain superficiel. And this was discovered also by the French, by Christian Jouberty and his students. And it's in English, it can either be called the superficial underground compartment, which has the wonderful acronym of SUC, or somebody suggested mesovoid shallow substratum. Most of us just avoid it and use its initials or use milieu souterrain superficiel. If you look at the middle picture here, which says classic MSS in France, you can see that this is the habitat where it was first described from, and it is a mixture of rocks and dirt, and the animals live in the interstices, particularly around the rocks. Now, the reason they looked here was because they found species that were found in two different karst areas, two different cave regions, and they appeared to be the same species, and so they wondered how they got from one to the other. So originally, Jouberty, although less his students who actually were the ones that studied it, particularly Charles Gers, didn't think it was a dispersal corridor as much as a habitat in itself. So it was originally defined as basically a covered scree slope, but on the left you can see a scree or a talus slope, this one in the Czech Republic, and you can, if you look carefully, maybe you can see a figure there that's Milos Zaccarda clambering around on this scree slope. If you bury a pitfall trap, a baited pitfall trap, and I won't describe the way that you can actually first bury one and then also find it again, but it requires a fairly elaborate procedure, because if you just pick up a rock on a scree slope like this, another rock comes to take its place immediately. So anyway, we don't need to worry about the technical aspects of it. Ruzicka and Zaccarda, the two Czech biologists that studied this the most, left these traps in for 1 year. I can't imagine any American with the patience to wait for a year to find something. But when they did, they found mites again, but also beetles that were both eyeless or reduced-eyed. And so you have a subterranean fauna, even in this very harsh scree slope. On the right, you can see a picture of Tanya Pipon again in what's called clinker MSS, which is basically a volcanic MSS in a very harsh environment. And basically where she is sitting, you can sort of see this mixture again of soil and rock, and it also has a rich fauna. In some of the islands, in the Canary Islands, there are no caves, there are no lava tubes left, but there's still a cave fauna occurring in this MSS. Well, finally, we get to to something in English and something that was found first in the U.S. This is the diagram is one I stole from Gary Schindell's article in the Encyclopedia of Caves, and it shows the sketch of the Edwards Aquifer. And the fauna was first described from artesian wells, particularly in San Marcos, Texas. You can see that this was known really a long time ago, way back in the 19th century. And in fact, Rakovica himself knew about it. This is more like a cave. I mean, if you look at this picture, this is, of course, a representation, but in fact, there are certainly big open areas. They're just water filled. So it's really it depends on your definition of a cave, but it's a cave-like thing. It's impossible to get to by any means that we have, because you'd have to go down a wellhead. You can access this fauna by some caves, like Ezel's Cave in Texas, provide some access. You can see an example of sort of how that works on the upper left of this sketch. This is particularly in the Edwards Aquifer, it's a very rich fauna. Glenn Longley was the first person to highlight this fauna, and had a paper that said, is this the most diverse groundwater fauna in the world? And as far as we know, yes, because it has dozens of unique species. I think over 40, but I don't recall the exact number. Well, sort of this section finish up with what really is a cave or caves, and these are these remarkable caves in iron ore in Brazil and probably elsewhere. And they're remarkable. They're formed by the microbial action of iron-reducing microorganisms. So they're sort of biological in origin. Seth Parker, a student of Hazel Barton, was the first person to really puzzle this out. One of the things that's interesting about it is the mean length of these caves is 30 meters. These are great caves for elderly cavers like myself, because you can say, yeah, I saw it all. It is the rare iron cave that actually has a dark zone. Here you see a picture of one of the big ones with a survey. And the fauna there is probably not, if it is just in these caves, it's mostly in a lighted area, and I'll get back to that in a minute, or it's also found in the MSS in this area, which seems to be more likely. It's just that caves are a convenient place to collect the organisms, and probably there's a greater concentration of organisms there, perhaps because more organic matter comes in there. So these are the iron ore caves. It's a very rich fauna. It has a number of really interesting and difficult management issues because it's not near iron ore. It is in iron ore, and iron ore is the main economic muscle of Brazil. So in the so-called iron triangle, and a lot on the edges of active mines, you will find a lot of these caves. So how do we make sense of this variety of non-cave habitats? We can just sort of tell stories and make lists, or we can try to sort of classify them in some ways. And it seems like there's at least two axes along which we can organize these habitats. And the first one is by pore size. And Bortolaniano, who wrote the book called Stegofauna Mundi, the stegofauna or the aquatic fauna of the earth, which is a great title. He split habitats into two groups, permeable and large and permeable and small. Permeable and large are caves and tunnels and deep aquifers, and the small ones are, of course, things like, he was talking just about aquatic things, were habitats like the Hypotelminorheic if you remember that from the beginning. And Tanya and I suggested that depth was important as well. We thought depth was important because shallow habitats differ from caves and other deep habitats because they have more connections with the surface, they have more environmental variability, and tend to have more organic matter. Well, we can ask the question, what unifies and what differentiates shallow subterranean habitats? And what unifies all of these shallow subterranean habitats is the absence of light, multiple surface connections, elevated organic carbon because of those surface connections, and with more environmental variability, which is often cyclical. And they differ particularly in habitat size. So there's larger and smaller ones, and so this is back to Bodhicianianu's distinction. So there were a lot of features we felt that unified these shallow subterranean habitats. So what unifies shallow and deep subterranean habitats is, what do they all share together, and what's different about shallow and deep subterranean habitats? It's really a slightly different question than the one above, and really the only feature that all these subterranean habitats share is the absence of light. Well, in a couple of minutes I'm going to say maybe they don't even share that completely, and they are different. That is, shallow and deep ones are different because there are more surface connections with shallow subterranean habitats. There is more organic carbon typically in shallow subterranean habitats, and there's greater environmental variability. So this is at least the start of a differentiation of the different habitats. Going along with that is the question of what's the nature of natural selection in these habitats, and for both within shallow subterranean, deep and shallow habitats, they all are convergent with the absence of light. So that should result in convergent evolution. That is, for example, the absence of eyes, the absence of pigments. And they could be divergent among and convergent within habitats. So, for example, habitat size is different in different habitats, but within a habitat, it's the same. Organic carbon can differ, cyclicity can differ. And then finally, even within a habitat, there can be interspecific competition. We know this particularly from the work in Slovenia and Croatia, where there can be 5 or 6 species of amphipods in a single cave or in a single site, for example, a hyporheic site. We don't approach that diversity typically in the U.S. except in places like the Edwards Aquifer. So we can have convergence, we can have divergence in the subterranean realm, just as we can in any surface habitat as well. So there are several emerging patterns that I want to just briefly mention. One is body size variability. Another one is habitat specialization. Third one is the occurrence of eyeless, de-pigmented species in dim light. And we've already seen that for the iron ore caves in Brazil. And then finally, the idea of biodiversity hotspots, which we saw a little of when we talked about the Washington, D.C. Hypotelminorheic habitats. Well, if you look at body size variability, if you look at the graph on the right, the black bars are the size of mites in microns in different sizes in caves. And the open bars are the ones, the sizes of mites in soil. So we can see that, in fact, of course, soil has a smaller pore size that the mites tend to be smaller. There's a lot more smaller ones and very few large mites. Mind you, a large mite is not exactly a frightening creature. So what we think is happening is shown on the left. For small sizes, fertility selection, that is, the bigger you are, the more eggs you have, the more offspring you leads to an increase in body size. This is counteracted by the habitat itself. If the size of the pore is a half a millimeter and the organism is a millimeter, it's not going to work. So it can only be to the size that can be accommodated by the pores. But at some point, the species don't keep getting bigger. For example, if we're walking down a large cave passage, the beetles in that passage are not larger than the ones in a crawlway. So there are sort of structural constraints and phylogenetic constraints. Perhaps we should all be grateful, but there are just no beetles that are a meter long, or even 10 centimeters that are found in caves. So there's that constraint, and so the pattern is complicated. And I might just say parenthetically, there's probably nothing harder almost in ecology to understand than body size, because it's affected by so many different factors. Well, this is simply to show that the habitats themselves occur in different forms. This is particularly true the MSS. The MSS is just a name that we attach to all of these habitats, terrestrial habitats that are of sort of medium pore size. And here you see what's been called by Ortuno alluvial MSS and colluvial MSS. Alluvial MSS periodically floods because it's basically a stream bottom. So the species found there are going to be probably slightly different than the species found in the colluvial MSS. There are also bedrock MSS, just like what it sounds like, the breakdown of bedrock, and then ones associated with volcanic terrains. So we can see that sort of increased specialization of habitats. I mentioned that light was absence of light was a unifying factor. This is a map, and it's a map showing how much light occurs in a cave called Cueva del Muevelo Rico in Cancun, or near Cancun, very near Playa del Carmen. And we did a series of light measurements, and you can see the result of the light profile there. The black, the little piece of black, is the only place where there's true darkness. The gray, there was less than 0.01 lumens of light. The blue is from 0.01 to 1. The yellow is 1 to 100, and then the red is over 100. This cave is about a mile long, and it has, I think, over 15 entrances. So basically, any place you are in the cave, you can see light, except for that one little area in the sort of upper left of the cave, which is the only real dark zone we could find, and thanks to Peter Sprouse for actually telling us where it was. What's remarkable, well, there's several things remarkable about this. One was how difficult it is to measure with any accuracy light levels that really dim light below a lux. And that's perhaps another story, but we simply know that it's less than 0.01 lux. We don't know how much less because we were able to show photographically that there was light, but we don't know. We couldn't measure it with the instruments that we had. But what's remarkable is this is a big twilight zone. The cave fauna is the same as the fauna of a completely dark cave. And we think the reason is that there's simply not enough light, except right in those red spots and perhaps blue spots right by the entrances for any eyed predator to find anything. You know, 0.01 lux is like a very dark night with no moon and an overcast sky. You can detect the presence of light, but it's very hard for an organism to actually, I think, detect shapes. So this idea that what unites everything is a complete absence of light perhaps needs to be modified a bit because we have these. Now, I'm not sure we're supposed to call this a cave. I mean, is only the black spot in this area really the cave? You certainly have to wear caving gear and have a light to see this area, but I think you see what I'm trying to get at here. Well, the diversity of these sites can be very high as well. And this is a slightly busy and complicated slide, but what it's showing is the partitioning of diversity of the number of copepods that are found in epikarst. We sample epikarst by sampling dripping water in a cave. So that's water, that's epikarst water. Tanya Pepon did this in some caves in Slovenia for about, I think now 15 caves, sampled over a year. Once again, needing some patience. Water is all filtered, and then the copepods are identified and counted. Among these 15 caves, we found a total of 29 species that were eyeless and without pigment, which is really a remarkably high number. And in one spectacular drip, I mean, there's nothing more simple than a single drip of water, dripping water, that in that drip over a year, she found 10 species of animals. So if you look at the bar graph on the left, you can see that at the bottom shows us the average number of species per drip was 3. The number among drips within a cave, but different drips, was 2.7. So we have about 6 species per cave. Different caves in the same region added another 5, and then different regions added another 18 species. So it's a very rich fauna, and it's one that's very localized and rivals the diversity of the whole caves themselves. Well, what about the U.S. contributions? I've been talking a lot about stuff that's happened in Europe, and there's a couple ways that U.S. scientists have studied this. One is that stream ecologists, and there are many stream ecologists in the U.S., study the hyporheic but not cave-like animals. They're more concerned about water quality, nutrient exchange, and things like that. And the same thing is true for a whole range of soil scientists. So there haven't been, those habitats are studied, but not with respect to their subterranean nature. Now, the study of deep phreatic habitats, particularly the Edwards Aquifer, remains a U.S. strength, and there's really no other equivalent. The work of Benjamin Schwartz and Ben Hutchins now, particularly. But there's also there are 2 different world views that we can see about the subterranean habitats. And if you just look down at this last point, think about, many of you probably know the excellent books by both Art Palmer and Will White. Art's book is called Cave Geology. Will's book is called The Geomorphology and Hydrology of Karst Terrains. So Art focuses on the cave. That's sort of the central focus. Will is focusing on a landscape. When you focus on a landscape in a biological sense, then you move to these non-cave habitats. And it's kind of an interesting mindset that we can see from the late Tom Barr, who's a giant among cave biologists. He found a beetle, Pseudanophthalmus, of which there are over 200 species in caves in the U.S. And you can see the title of his paper, An Epigean Environment in West Virginia. If you read the paper, you discover that he actually is talking about an MSS habitat. That is a mixture of, rocks and soil not so far from the surface. Now, he wrote this paper before Jouberty wrote his paper about the MSS, but it didn't occur to Barr, who's very sophisticated in terms of understanding the terrain, that this was really a subterranean habitat. In fact, he calls it a surface or epigean environment, even though the animal has no eyes. So there's these two different views, and it's really the difference between speleology and karstology. And speleology is, for instance, well represented by Frank Howarth's classification of micro, meso, and macro caverns. You can see just the focus on spaces, and shallow subterranean habitats may be an intermediate staging habitat for these, but we have no evidence of that. Alexander Klimchuk, the Ukrainian geologist, emphasizes the primacy of speleogenesis as to understand not only caves, but to understand karst terrains as well. But karstology, a somewhat awkward but useful word, is the standard paradigm for the karst geosciences. And Raymond Rouche, a French biologist, used the model of a karst basin, that is the whole basin, not just the cave, as an ecosystem starting back around 1970. And this concept of the basin rather than the cave as a unit of analysis had a great deal of currency, has a great deal of currency indeed, particularly in Europe. And I'd like to finally just thank all the people that hosted us. Tanya Pepon and I got to see these habitats all over the world. This is obviously pre-COVID days for sure. And Louis Darwin and Anne Bedos and Paris Museum of Natural History, Charles Gers in the Pyrenees, Rijeka in Jakarta in Czechia, Slavko Polak for the MSS in Slovenia, Pedro Aromi for the MSS and lava tubes in the Canary Islands, Fred Stone and Bill Jeffrey for lava tubes in Hawaii, Florian Millard for wells in France, which I didn't really talk about much, Marie-José Dole-Olivier and Pierre Marmonier for the Hyporheic in France, and Augusto Auler for the iron ore caves of Brazil. And so my last slide is if you really would like to learn more, and let me try to sell our book, or not necessarily sell it, just get a hold of a copy in a library or elsewhere called Shallow Subterranean Habitats, and you can hear a lot more on this topic. So I want to thank you for your attention, and I guess it's possible that I'll get some questions. So Debbie, it's back to you.
Okay, thanks, Dave. All right, awesome. Well, thank you for this information. Interesting pictures and graphs. All right, now it is time. If you have any questions, please text them in. And I will then go ahead and ask Dave, who will be so kind as to share more of his knowledge and understanding. There are several webinars, though, that the presenters do such a phenomenal job that there seems to be no questions until after the webinar. Then a lot of people will then email questions, and I'll ship them off to Dave. Does anybody have any questions at this point you'd like to ask Dave that I can go ahead and relay to him? If not, what I'm going to do is I'm going to thank Dave very much. I appreciate this. I know you're busy, and I appreciate you doing this at this time. The lockdown, which we can't go caving, and our members are getting cabin fever. We really appreciate the fact that you're willing to share this. It helps a lot, giving education and information, which is part of our mission with the NSS. So on that, Dave, I'm going to thank you very much. Thank you, everybody, for appearing and listening tonight. All of the webinars, including this one, are recorded and stored on the NSS website of caves.org. You can find them, download them, or watch them. We encourage you to share them with your grottos, your regions, any educational reason that you would have. If you have any ideas on what you think would be a good webinar, or if you have good information that would be good for us, please let us know. Everybody have a safe night. Be careful out there and cave safely. Till next time, goodbye.
Goodbye.